So next up is Christopher Vulpe from the University of Florida, who will be telling us about genomic screening to examine gene-environment interactions.
Okay, thanks a lot. Okay, the green button. Is this the green button?
That's the top one.
Top one. Top one. Okay, the big one. Okay. Okay, well, thanks a lot. I'm going to tell you a little bit of a story about our efforts to use, to take advantage, I guess, of the tools that have been developed by other people and applying it to toxicology relevant questions. And we've heard a little bit previously about individual knockouts, and I'm going to tell you today about our efforts using whole genome screening approaches. There's a large number of people involved in this work, and Amin Sobe, who is a very talented graduate student in my lab, has been responsible for essentially all of the work that I'll present in collaboration with several people. So there was a question by, I believe, Lisa, about, well, how the heck do we put this into a context that's useful for a risk assessor? So I very quickly inserted a slide or two to maybe try to address this question, which is, and you know, when I think of adverse outcome pathways, I think in terms of a cell, and so this would be focused on an adverse outcome pathway in a cell, or a cellular adverse outcome. So how can we use CRISPR and these approaches to try to understand what is a toxicant doing and how is it leading to its, as very cleverly, as was put to us before, the bad stuff. So here is my generic cell, and I think what you can see here is that there's a whole series of steps steps where the toxicant is brought into the cell, it's metabolized, maybe it hits something, there's a bad outcome here, the cell responds, maybe there's another one up here and sort of responding to that. And our challenge is, for a given toxicant and for the vast number of toxicants, how the heck do we figure this all out for all of the cells out there? And my thesis is that by actually getting rid of genes, we can actually find out of all of these genes that are present in the cell, we can start to tease this apart and identify the genes that are important in the response to the toxicant, and maybe sort of work backwards in a reverse genetics way, work backwards and figure out, well, here are the genes that are involved, and help, because basically you get rid of it and it causes the cell to croak, you say, well, it's probably important. Well, we don't know exactly how it's important, but at least it gives us a clue that it's involved. And so what we can do then is build this pathway up slowly to build these adverse outcome pathways, or a cellular adverse outcome pathway, which then potentially could be used more in a risk assessment framework. So how do we do this? Well, we heard just now about the idea of using an individual knockout. And so this is certainly a very powerful and useful technique. And the idea here is that you target an individual gene that you're interested in and use the technology such as Cas9. You then isolate that cell or organism, and then you can assess in a very targeted way, as we saw with AHR, what the effects are. The approach that I'm going to talk to you about today is using an alternative approach, which is basically simultaneous targeting of multiple genes at the same time. So it's essentially the same idea, but what you're doing is that you're targeting a single gene in an individual cell, and you're making a pool of mutants. So each cell has a different mutation, and you can then assess the function of all of these genes simultaneously by looking at this pool. And the easiest way you do it is you just screen for sensitivity to the toxicant through whatever assay you happen to like, and to identify and then rank the important genes. And this is the approach that we have been using. So this is just an illustration, and this is actually an illustration that I used back when we used to work on yeast. and we still work on yeast. And yeast, as you heard, had this tools and technologies for a long time. And I don't know how many times I was ragged for using yeast and not using a mammalian cell, and you really should do this in a mammalian system. And it's like, well, the technology. But luckily, I was at Berkeley for a while, and Jennifer Doudna was there, and she probably heard all these complaints, and so she very kindly sort of transformed biology so that we could take advantage of this technology. And I was very thankful that she was willing to do that for us. And so the idea here is basically the same idea as what we could do in yeast a long time ago. Now suddenly we could do with mammalian cells. And what you could do is you grow them, you expose them to your abuse of choice, whatever it is, and every gene, and what I didn't point out though, is that every knockout is not only is it knocked out, but since the vector that you're using is integrated into the genome, it acts as a barcode for itself. So not only is every gene knocked out, but it's individually barcoded. So you can go back and ask afterwards, after you've grown them, you just collect them in a pool and you just count these barcodes. And if nothing happens, then you say, well, it's not probably important. But if it grows really poorly, then you say, well, this is probably important. And if it grows well, you'd say, well, it's probably important as well. And therefore it can give you ideas of what's going on. Okay, so I included this because there were some caveats that are involved in these whole genome approaches. And I thought for this audience, I would sort of try to point out some of these issues and do this preemptively before I sort of talk about the screens that we've done. So essentially, we're using an in vitro approach. So we are open to all of the problems and issues with using an in vitro system, which is metabolism sucks, we're using immortalized cells, and we've got screwed up toxicokinetics. So we acknowledge those as major issues. Now, you can target every gene in your library, but it's important to point out, and I think I sort of said this, you're only looking at a single gene in each cell. It's also important that we're not looking at homogeneous knockouts. We're looking at a heterogeneous population of knockouts. So these contain a knockout of a single gene, but each gene is knocked out in an individual cell in a different way. So it's kind of a heterogeneous pool of knockouts that you're looking at. The genes are knocked out on each chromosome, but in general, it's not going to be the same knockout on each chromosome. It's going to be a different knockout. And so again, you're looking at a heterogeneous pool. They're all tagged, and we are using it now there are many different assays, but we're using a relatively simple endpoint at this case, which is just its growth advantage or disadvantage in response to an environmental exposure. But you could certainly think of a lot of other endpoints that you could look at. So as was pointed out earlier, the initial efforts in whole genome screening were focused on application in cancer very appropriately. And until recently, there's been very limited number of studies looking focused on toxicology. And they were pointed out earlier, there were 2 on triclosan and paraquat. But all of these these are basically toxicants. And so I think that you can get a lot of information by studying many of these studies on chemotherapeutics. Okay, so what have we done? Well, we focused on 2 prototypical toxicants, acetaldehyde, which is an oxidative metabolite of ethanol. It's a genotoxicant, a carcinogen, and it, although there's controversy, may be associated with alcohol-associated cancers. And unfortunately, even though it's been studied for a very long time, the mechanisms of toxicity are still very poorly understood. Similarly, we worked on arsenic, and we had done some work in arsenic previously in yeast, and we worked on a particular version of arsenic, which is arsenic trioxide, which is actually metabolized to the usual metabolites within the cell, MMA and DMA. It's also a carcinogen. As you know, there's drinking water exposure. And arsenic, I'd say, is the poster child for diverse mechanisms of action and is extremely controversial over what are the mechanisms by which it acts. And so we thought, well, we don't, in a way, we almost have too much. There are too many different mechanisms out here. We don't know which ones are important. And could we use this whole genomes approach as a way to maybe rank these mechanisms or get some insight into what's happening and guide us as to their relative importance. So how did we do this? Well, we use this as now sort of a standard approach. So you infect lentivirus with your libraries, and we used a gecko library, which is a library not from a gecko, but is the name of the library that was made and is available from AdGene. It had 18,000 genes. We exposed them to our toxicants at doses that lead to about a 50% decrease in growth. And they basically collect the cells that survive and count them. And as I pointed out, the hope is that we would identify some resistant mutants that will be enriched in the presence of toxin, as well as sensitive mutants. So what do you find? Well, for people who have looked at RNA-seq data, this looks very similar to RNA-seq data. And this is showing here the number of counts and the relative exposure, the relative frequency of that mutant in the pool as compared to the untreated. So this is looking at the relative abundance of an individual mutant cell in a population of the mutant cells in the pool. And so if there are more of that sgRNA knockout, then it would be considered resistant and would show up here. And if there are less of that knockout gene, then it would be considered sensitive. So it's very similar to RNA-seq. There is a twist in that we generally use, as was pointed out before, that there are the computational algorithms that are used to predict where an sgRNA is going to bind are imperfect, and therefore we use multiple sgRNAs to target every gene in the hope that if we see the same thing with the multiple sgRNAs, that it's probably the effect that we're seeing is due to the on-target effects rather than the off-target effects. So ultimately, we measure the mutant cell abundance in the pool. And when we did this here for the arsenic, we identified candidates for a secondary screen. So we did a two-step process. We did a primary screen with a whole genome, and then we went on to do a secondary screen. So here is the outcome from the primary screen, and the data you get is very similar to the data you get for an RNA-seq. You get the gene, obviously, and then you get a log fold value, which shows how enriched or depleted your mutant cell is in that population. So in this case, what this means is that knockouts of KEAP1 are enriched in the population. And so knockouts of KEAP1 are resistant to arsenic trioxide. And similarly, these genes, which I'll talk later, involved in selenocysteine metabolism, are enriched in response. Then there are also other genes which are actually sensitive. So as I said, we did a two-step process. We did a whole genome to identify candidates, and then we said, well, we got a whole bunch of candidates. How can we figure out which ones of these are real and which ones are not? So we did a secondary screen where we actually focused, where we made a custom library. We made new genes, and we identified the candidates, and then we ran the screen again, and then saw how reproducible. And again, we saw the same sort of thing. For instance, KEAP1, again, for 8 out of 8 sgRNAs, there was a significant enrichment. And similarly, we saw sensitive. Okay. So we already knew this to a certain extent about KEAP1. So KEAP1 is a well-known inhibitor of NRF2, which is the primary mediator of reactive oxygen response to reactive oxygen stress. So getting rid of KEAP1, not surprisingly, increases the resistance to arsenic. So we knew this already, but it suggests that it's very important in the acute short-term toxicity that we're looking at. We found that there were a series of transporters that seemed to be implicated. We found that getting rid of the aquaporin 3 gene seemed to increase resistance, and getting rid of the MRP seemed to increase sensitivity, which suggested a role that these potentially play a role in transport of arsenic, although that needs to be directly demonstrated. And then most surprisingly, what we found is that loss of a whole series of genes involved in selenocysteine metabolism were, when they were absent, actually increased resistance to arsenic. And so this was really surprising to us, and we are still trying to figure out what it is, what's going on. And this is just showing a validation where we actually made an individual knockout and showed that, in fact, individually knocking them out also increased resistance to arsenic. So this was an unexpected insight. And I think what's important to point out is it's not just one gene. It's essentially, if you knock out any of the genes that are involved in the selenocysteine pathway, it increases the resistance to arsenic, which really suggested to us this was probably not an artifact, and that it really is clearly something involved in selenocysteine metabolism. So what's our current hypothesis? Well, we have several hypotheses, and I'll just say one, which is that it's been well known for a while that there's an actual potential physical interaction between selenium and arsenic. And it's one of the rare cases where you can prevent selenium poisoning by giving people arsenic. And you can prevent arsenic poisoning by giving them selenium. I'm not suggesting you do this at home, but this is actually possible. So we think that this, which has been known in whole animals for a long time, we think this similar mechanism may be going on within cells. And so when we knock out the selenocysteine metabolism, we're actually increasing the intercellular pool of seleno compounds, which allows the formation of a selenocysteine complex, which is potentially less toxic and then possibly exported. And we've actually now validated this by actually just manipulating selenium levels, and in fact, you can affect the toxicity in response to arsenic by manipulating the selenium levels. So in summary, this is what we've found, and given the relative limitations of time, I'll just give a very brief talk, a very brief introduction to how we've applied this now to acetaldehyde. We did a very similar thing, and these are the results from the secondary screen. And what we found, again, validated what was previously known, in that DNA repair seems to be really important. And specifically, it appears to be, whoops, how do I go back? It appears to be transcription-coupled repair, which makes sense to a certain degree because it's thought that acetaldehyde forms addicts, and that the removal of these addicts requires this mechanism of repair. But we also found something completely new, which is this gene which we called OVA-CA, which is called OVA-CA2, and it's the ovarian tumor suppressor candidate gene. And we don't know what the heck this gene is, and nobody really has any idea what it is, but this suggests that it probably plays an important role in this process. And I'll just give you a little taste of the data that we've just recently done in a collaboration with Alicia Stornetta and Sylvia Balbo, is that if you knock this out, and we did this in an individual knockout, we actually affect the production of acetaldehyde, not production, the amount of acetaldehyde addicts that are present in the DNA. So in the absence of this gene, the number of addicts increase in the DNA of the exposed cell, suggesting that these play some role, which we don't know yet, in the removal or the at least in the removal of acetaldehyde addicts from the cell. So finally, what have we learned? Well, I'd say that it's given us some, it's reinforced that oxidative stress is really important. It's suggested maybe not completely unexpected in retrospect role for selenium metabolism. It suggests that DNA damage is really important in acute aldehyde. We've got a new gene which we have no idea what's going on. How can we use this? Well, my hope is that although any one screen isn't going to tell you everything about the process, I think it helps fill in some of the roles, some of the holes in our knowledge, and that the use of this can help us sort of work out these adverse outcome pathways. And finally, I just want to give one shout out to a new approach that we're using with Tren Liu and Luping Zhang, which is we've made now a targeted library that's focused on toxicology-related genes. We stole liberally from the Tox21 program, took all of their genes. We added a whole bunch of genes from the environmental genome. We weren't satisfied with that, so we grabbed a bunch of random genes that we thought were important. We made this library, which now has 3,000 or so genes. And the idea here is that this would be enriched in things that are of interest to toxicology and have previously been shown to play a role in toxicology. And this is just showing all the processes that are involved. And maybe this could be a way to use this sort of approach in a more rapid, maybe a more focused approach. And then finally, I'd like to thank all of the people who've been involved with this. As I mentioned, this work was done primarily by Amin with the assistance of Manny, and who's been involved in the Tox CRISPR primarily, and Alex is our bioinformatics. This is a long-term collaboration with Liu Ping when I was at Berkeley, with Martin Smith, who was the PI of the grant, worked with Tren Liu on the Tox CRISPR approach, and then most recently our collaborators from the University of Minnesota who have done the acetaldehyde toxicity, the acetaldehyde addict work. And of course our funders from the UC Berkeley Superfund and the NIEHS. And with that, thank you very much.